Welcome to the Blueprint of Life video 10. In this video, we're going to be discussing chromosomes and how the work of Sutton and Boveri have contributed to our understanding of chromosomes. So here we have a quick infographic that is trying to connect all of the different people that we're learning about and how in fact we come to our genetic understanding that we have today. So we've learned previously about Mendel and his work on his pea plants and how we looked at the inheritance of traits, specifically those alleles and how they are inherited from through generations. And now in this video we're going to be looking at the work of Boveri and Sutton and how they both independently contributed to what we understand today as the chromosome theory of inheritance. So Boveri studied the behavior of sea urchins and how their chromosomes behaved during meiosis and after fertilization. So he looked specifically at the cell nucleus and chromosomes during these processes, and he used sea urchin eggs because they could be easily fertilized in a lab and that the larvae developed in 48 hours, which allowed him to view these changes during meiosis. And Boveri's work showed us that in the nucleus of the egg and the sperm, egg and the sperm, that they both contribute the same amount of chromosomes to the zygote, which is the fusion of the egg and the sperm, and they both contribute 50%. This was the connection that he made between chromosomes and hereditary. And then Sutton performed some work on grasshoppers, and he also studied meiosis in their cells. And he produced a connection between the behavior of chromosomes during meiosis and the laws that Mendel had proposed in his laws of heredity. His findings, combined with those of Boveri, formed our chromosomal theory of inheritance. So Boveri specifically showed us that the offspring inherited their characteristics from both of their parents, and that in fact a complete set of chromosomes is needed for development to be normal. He found that the factors of inheritance that Mendel described were found on chromosomes that are held within the nucleus of cells, and that there are more than just the inheritance of chromosomes and the many factors. There is more than just one on each chromosome, and that there are in fact many factors and genes. Sutton, however, he described that chromosomes occur in distinct pairs that he could visualize during meiosis, and that these pairs, one were inherited from the mother and one from the father, and that pairs were in fact the same size and shape, which we now call today as homologous chromosomes. During meiosis, the number of chromosomes separate in a cell, and each gamete receives one from each pair, and then after fertilization, the full set of chromosomes is restored. Here we have a quick summary of scientific understanding and how it changed before and after the work of Sutton and Boveri was realized. So previously, they thought that hereditary factors were found in the cytoplasm and the nucleus, but Sutton and Boveri showed us that it was in the nucleus only. We weren't quite sure about where the material was stored, and in fact, Sutton and Boveri both showed us that it was on chromosomes. That these factors that were discussed before Sutton and Boveri's work were passed on to the next generation from random assortment during meiosis, and that the nature of chromosomes in fact didn't disappear and reappear as some scientists had suggested, instead that they occurred in pairs and each pair of chromosomes was homologous. We know that chromosomes are compact coils of DNA molecules and they're organized around proteins. The only way that DNA can fit into each cell is if this occurs. And that each gene is made up of a portion of DNA and it stores the information as a coded sequence, and that each gene is held at a particular site, which we refer to as the locus on each chromosome. So therefore, a chromosome is a linear sequence of genes, which is why we say that there are many genes found on one chromosome. Here we have a quick diagram of how, from the smallest to the largest bits of genetic information that our body holds, and we can see that this supercoil and this winding that occurs is really, really crucial to ensure that all the information is stored without taking up too much space. Thank you for watching video 10. Make sure you tune in for video 11.